So it's a real pleasure to be here with you. And maybe if I could have a clicker. I thought it was here, but it wasn't. So I'm going to start by telling you, and also if you could move to the first slide. Thank you so much. So we're going to talk about design and the life sciences. Now, of course, we were saying how, OK, first slide. OK, thank you. Michael Craig Martin was saying something wonderful about these chairs, and he was saying that these chairs are perfect, and God bless him for breaking a spear for design. But even more perfect is that ranchu goldfish. It's a fish that was initially coded by a divine spirit or by the beginning of nature, or however you want to describe it, but then was refined and redesigned over centuries by human beings. And science and design are forever intertwined. They're so connected, they are inextricably linked. You see, this is a new work by Neri Oxman. It's a Krebs cycle for the metabolism of creativity. And it's a little too dense to explain in 5 minutes, but maybe you could take a note of it and research it later. It shows how art, science, engineering, and design contribute to make the best of our built world and how linked they are. And when it comes to design, one of the biggest metaphors for science and for creativity has been nature, forever, throughout the centuries. The biological sciences, the life sciences, had given designers so much fodder for creativity. And you see here just a few examples from Cuvier in the 19th century to Leonardo da Vinci, and then the quark cell structure by Robert Hooke, who was one of the first to really use microscopes. But forever, there's been this link between science and design, and in particular between the life sciences and design. The life sciences is a relatively new term that is an umbrella term for all the sciences that study living microorganisms and organisms, ranging from biology to biotechnology and even biodesign. But the metaphors for design in the past tended to be very natural, the imitation of the forms of nature. Today, that imitation also entails using the computer to get as close as possible to the metabolic and the creative methods of nature. So we go from Guimard to Neri Oxman and Joris Laarman. And more than ever, I discovered that when I did a show in 2008 called Design and the Elastic Mind, which was a show linking design and science. At that time, I came to a realization that designers and scientists are made for each other, that they should meet and work together without too many limitations and too many filters. And I also realized that designers are the enzymes for innovation. Revolutions might happen in science, but without designers, these revolutions would never become life. Designers take microwaves and help them become into ovens, or they take the internet and from lines of code make it into an interface that people can use. Therefore, design is really fundamental in its link to science. And when I worked first on the exhibition, Design and the Elastic Mind, in 2008, we then went on at MoMA to also write the first book about biodesign. That was by William Myers, and you see it up there. But slowly but surely, along the years, biodesign, the design that uses not the materials of yesteryear, not wood, not plastic, not the concrete of architecture, but rather living tissue and cells, has become more and more acknowledged to be a very important field for the future. Of course, there's been a lot of small niche applications of it. You see, for instance, a quite beautiful panel that we had at MoMA just 2 years ago that was based on the book Synthetic Aesthetics, of which Daisy Ginsberg was one of the editors, and also the Biofabricate conference that happens every year in New York City that gathers designers and scientists to discuss in vitro tissues and the future of biodesign. But recently, there was also an article on Wired magazine that talked about the fact the title was kind of very loud, said, "Move over, Johnny Ive. Scientists and biologists are the next star designers." So it's becoming more and more acknowledged that design and science, and especially biology, come together. One of the first instances when I understood that was at the exhibition Design and the Elastic Mind, when I showed this little coat made of mouse stem cells that was designed by Symbiotica, a wonderful lab out of the University of Western Australia in Perth. It was a living organism that was shown in a design exhibition. To me, it was quite an epiphany, and it was the same also for the public and for many designers. And from that moment on, I've been trying to gather examples of design that not only imitates living organisms, but actually incorporates them in the design. This is another beautiful work by Neri Oxman out of the MIT Media Lab, she who designed also the Krebs cycle that I showed you before. And it's a pavilion that's been designed so that silkworms are the actual construction workers. So everything is set up so that the silk that they will create and fabricate will constitute the pavilion. Or another example that I can bring to you of an architecture actually made using living organisms is this tower that was at MoMA PS1 2 years ago in the Young Architect cycle that was made using bricks that are made of corn stalk kept together by mushroom mycelium, cured and kind of grown in a factory nearby. And you see it's completely biodegradable, and still it's a structural element. In the book Synthetic Aesthetics, Drew Endy, who's one of the most well-known synthetic biologists, actually biologists, and one of the fathers of synthetic biology, talks about the role of designers in this new burgeoning field. In particular, he says that to view synthetic biology as merely being the domain of scientists and engineers is a mistake of the first order. He says you don't have to be a scientist or engineer. We're all synthetic biology together, and design is part of this cycle. It's something that is very important because what design is, the definition, is as fluid as many other principles today. This virus that Autodesk developed with software is actually part of the MoMA collection. It's a virus that is built in a different way and that is meant, like many new biological entities these days, to help with cancer research and with the delivery of medication. But the same scientists that developed it also spoke about the fact that this virus could be hijacked in dangerous ways. So ethics is also a part of the collaboration between designers and scientists, and a very important one. There are new projects that are ethically fraught and nonetheless extremely interesting. Using biology, Stuart Brand and Ryan Phelan are hypothesizing the possibility of bringing back species that have been extinct for a while. They're particularly taking the example of the passenger pigeon that used to be the most well-known and frequent bird in the United States at the beginning of the 20th century and is now completely extinct. And they're actually testing the possibility of bringing it back. Or even more wonderfully, this completely hypothetical project by Ai Hasegawa that is about creating children that come from a couple of gay women. And we know that it's very difficult for that to happen without a male counterpart coming in between the two women. And instead, she hypothesizes the ability of having a spawn children, of course girls, that come from that couple. She made a whole beautiful project and video about that. So science and design talk to each other, and they really come together in order to create new futures. And today we're here, I'm introducing this panel. A scientist and science expert is going to talk about it. John Gorman has devoted his life to stimulate an understanding of the relationship between the arts and the life sciences, and he will make it happen here in Munich with the new museum that will be built in the adapted part wing of the Nymphenburg Palace in a few years. And Daisy instead will talk about it from the designers and researchers' viewpoint. So it will be quite interesting because our goal is to see how these new ideas can be disseminated so that we can really bring design and science together. I'm showing here a kind of visual of a quantum computer because in a way we're all floating in this idea of ambiguity and ambivalence and trying to bring the whole world with us so that we can design a better future. So I would like to call to the stage now Michael John. Here he comes, Michael John Gorman. Let me help him say hello to him. Thank you. There you go.
Thank you, Paola. It's great to be here. I want to ask the question, if I can click. There we go. I saw this poster last week when I was walking through London. Funnily enough, I was on the way to the Serpentine Gallery to see Michael Craig Martin's beautiful exhibition. And I was intrigued by it. Where do you go to ask questions about life? And ironically, even though this poster was right beside the biological powerhouse of Imperial College, and also right beside the Natural History Museum, this actually suggests that the place to go to find out about life is an Anglican prayer meeting. We are experiencing seismic changes right now in the way we understand, explore, and manipulate life. And I believe that it's critically urgent that we find new spaces and places where people from all sorts of different backgrounds can go to find out what's going on and to be part of this revolution that is occurring in the life sciences. And I think right now it's very challenging. Where do you go? Back in the 17th century, this was where you would go to ask questions about life. This was the Wunderkammer of Ferdinando Cospi in Bologna in 1660. And here you could see all sorts of amazing creatures gathered from around the world, from expeditions, and even some hybrid creatures, probably created by Cospi himself, winged fish and so on. And Cospi lived in a world where the dream of having all of creation under a single roof was a plausible project. You could gather all of nature, all of life in a single place. And we've moved in the 19th century to a dynamic notion of life, first with Lamarck, then with Darwin, from this static idea of life. And right now we are experiencing the moment of moving. And Daisy Ginsberg will be talking about Pollination. Was the century of physics, then the 20th century of biology. Why? Well, it's not just about these large-scale projects like the Human Genome Project and the Human Brain Project now going on in the rise of a new species, the rise of the biohackers, the rise of the community biolabs, the DIY bio-community. And I believe that we're experiencing something very like the moment of Bell Labs in the 1960s, the explosion of creativity around art and technology that happened then, and also very like the moment of the Homebrew Computer Club and the hacking movement in the 1980s. It's happening right now for biology and the life sciences. Naturally, I'm biased towards Irish examples. This is the invention of an Irish biohacker called Cathal Garvey. And it's called a Dremel centrifuge. And Cathal Garvey he couldn't afford to buy a lab-quality centrifuge, so he designed a 3D printable attachment that you could put onto a Dremel drill. So a centrifuge that would normally cost thousands of dollars suddenly was available for under $100. And this allows you to do DNA extraction and all sorts of synthetic biology work from your bedroom. And in fact, he really did it in his bedroom. I think we are seeing an explosion of new types of spaces, often at the fringes of traditional institutions, such as community biolabs like the GenSpace in New York, and also events like iGEM, which bring together people around synthetic biology. And it's a very exciting moment because we haven't even really figured out the language with which to talk about this. We have the large labs, the biohackers, we have the FBI getting involved, we have security. Confusion of languages. But I think it's really critical that we do find a way to talk about it and bring particularly young people into the conversation about where we should go with these new technologies. One project that I've been very involved in is the creation of Science Gallery in Dublin, as Paola mentioned, which is all about bringing together scientists, artists, and designers to explore new life. And this is an example of one project there which involved a cell line from a girl, a 13-year-old girl, who died in 1969, which is alive in the gallery. And this is an installation which provokes all sorts of questions. Is this alive? Is this her body? How do we feel about the idea that we can buy her cells easily off the internet without her consent? So these kinds of conversations are very important ethical conversations. I want to give you 3 examples of projects where designers, who I believe are crucial for framing these conversations about new life, have helped to point a finger in the wound around new life sciences. One is from an exhibition we have on right now in Barcelona, in a beautiful place called the CCCB, called Human Plus, the Future of Our Species. And I strongly recommend to go and see it if you're in Barcelona until April 2016. The other is from an exhibition called Grow Your Own, which we developed with Daisy Ginsberg, who will speak next, which was looking at life after nature and synthetic biology from the point of view of designers. The first project is in Human Plus, and it's a beautiful project called the Human Pollination Project. And it's created by a designer called Laura Alcorn. And the idea is, imagine if we could no longer rely on bees and insects to do the work of pollination, and instead humans had to take over this job. And Laura Alcorn designed all of the instruments and tools that people would need to use to pollinate plants in place of insects. And they're really beautiful, they're silver, but they really, in the moment of colony collapse disorder and neonicotinoid pesticides, these really bring home the futility of attempting to substitute the ecosystem services provided by bees and insects. So it's a very powerful way to engage people with this question. Another project also in this Human Plus exhibition, which looks at human enhancement from prosthetics to the singularity, is called Foragers. And it's by 2 wonderful designers, also in the spirit of critical design, called Dunn and Raby, Tony Dunn and Fiona Raby, now based in New York at the New School. And Foragers posits the idea that, what are we going to do when we have 10 billion people on the planet, there's not enough food? Maybe humans will actually have to take a lesson from the cows who have 4 stomachs, and maybe humans will have to develop external stomachs so that they can pre-digest food. For example, pond scum or grass. And so this new culture of foraging where we can eat previously indigestible materials. Again, a very provocative piece of design that helps to ask questions about what future do we want to live in. And the final project that I want to mention is a project created by a wonderful scientist called Christina Agapakis and a smell provocateur called Cecil Tolas. And they collaborated together. This is actually in our community biolab in the Science Gallery in Dublin, to do a wonderful project called Self-Made, which was to create cheese using bacteria taken from prominent individuals. And we had, you can see some of the cheese here. We had a human cheese and wine reception, where you could, it says the wine is normal, and people were allowed to smell other people's cheese, but you could only eat your own cheese. That was important for health and safety reasons. And 2 of our very generous contributors of bacteria one of them is with us this evening, Hans-Ulrich Obrist, the curator of the Serpentine Gallery, who gave us bacteria from his nose to create this wonderful blue cheese that you see here. And then Olafur Eliasson, fantastic artist, his tears provided the bacteria for the other cheese, marked Olafur. This is a way to engage people with the conversation of the human microbiome, which you will actually hear about right after this session, and the question of humans as habitats for bacteria. I was really disappointed last week. I used to always love the scientific fact that there were 10 times as many bacterial cells in us as human cells, but last week they announced in Nature that, in fact, it's exactly the same. We have as many bacterial cells as human cells. It's a bit disappointing. But this brings home the question of the microbiome. So, a new museum of life sciences for Munich. Well, this certainly doesn't look like a new museum for life sciences. This is the beautiful Nymphenburg Palace located right outside Munich city center. And we're now in the process of designing a museum not a museum of natural history, but a museum of natural futures for Munich. And this is going to be located at this wonderful site, and it will be a place where we seek to reconfigure the relationships between humans and other species, involving designers and scientists. And the University of LMU, this is where Bernd Frankenstein was an alumnus, appropriately enough for looking at the future of life sciences. Thank you very much.
Back in 2009, I turned up at her office at MoMA carrying this briefcase. And I was on my way up to MIT to the International Genetically Engineered Machine Competition, or iGEM. It's a student synthetic biology or genetic engineering competition that last year was 2.5 times the size of this, so 5,000 students from around the world. But back in 2009, a team of students from Cambridge University were busy designing this, bacteria that produce different color pigments. And with another designer, we worked with them to start thinking about the implications of this invention as they were designing it. One of the things that came out of a workshop was this idea that maybe by 2039 it would become common to go to the supermarket and buy a probiotic yogurt drink that would contain engineered bacteria that would be able to detect for the chemical signals of different diseases. And if they detected something, they'd start producing the corresponding colored output, which was the contents of our briefcase. I took a suitcase of colored poo to meet Paola, and it was a very memorable first meeting. But this isn't a real technology, it's a fiction, a prop that we made to take to MIT to talk to the scientists about things that we saw that were problematic about this science. And from the perspective of a designer, the main thing for me was this idea that biology and life with it is being transformed into a 21st century design discipline. And as a designer, my questions have been, what kind of design discipline could this be? What should it be? And what is the role of design in biotechnology itself? Now, if you think this is all just science fiction, everything we've been showing you is far off future, I was amazed last year when a paper came out from Harvard University where they made this, programmable bacteria that can detect and record the signals in the mammalian gut. This was basically the first step towards making what we'd imagined real. And crucially, this research was sponsored by DARPA, the American Military Research Agency, as part of a funding call to protect American intellectual property in an emerging global bioeconomy, amongst other things. And it's now speeding up even faster. You may have heard of CRISPR-Cas9, which is a new kind of gene editing system that's completely transforming the field in months. And, I mean, Wired last summer described it as the end of life as we know it. This may be the case, but I actually think it's more like this at the moment. Synthetic biology is busy industrializing biology, using this technology to turn sugar into things that we already have, like ingredients for fragrances and flavors for food, and even the sort of far-off dream of jet fuel is something that's been abandoned. What's promising to be a disruptive technology is promising to do this without disrupting anything. And I believe as a designer who's interested in the future and how we shape things, this question is so crucial. It's what future do we want to design? And I believe design can be used to actually open up that question and shape a technology as it develops. And there's two ways I think that we can do this, or many ways, and these are the ones that I've experimented with. One is using design to imagine alternative futures, better futures, and the other is to actually question how we define better, to use design to actually understand the values that are shaping the technology. So I'll give two examples, and Paola mentioned synthetic aesthetics. And I just want to say that this was a project I was involved in for 4 years between Stanford University and Edinburgh University. And what was so unusual is this was funded by the National Science Foundation and the EPSRC as science. So we were able to bring in 6 artists and designers and 6 synthetic biologists all around the world in residencies, both in the lab and the studio, but it was scientific research. And that was so extraordinary to be able to bring these minds together, to crash different ways of working, to be able to ask and innovate around new questions and problems around the science. And the self-made cheese was actually one of these projects. But what we'd asked our scientists and artists and designers were these really obscure questions. Can we design life? How might we design life? And what might it mean to design it well? How do we measure that? So the outcome is in our book, Synthetic Aesthetics. And this is a disagreement between 20 people. I mean, I don't think any other book has so many arguments in it. And that's what was so extraordinary, is this is a conversation that has no right answer. There isn't a single way to say what's right and wrong about designing living things. But I think we all need to have that conversation together. And I'll just finish with this last conversation, which I think is the most important of all. And this was a conference that happened 3 years ago when the conservation community and the synthetic biology communities met for the first time. And they wanted to talk about whether, do they have anything in common, or are they actually going to be opposed to each other? Is there a conflict that's about to happen? And I drew this diagram while I was at the conference because it really struck me how the conservationists are busy looking backwards. They're pessimists and they're trying to stop the influence of humanity. Meanwhile, the synthetic biologists are optimists and they're reaching forward into the future, trying to design organisms for the benefit of humanity. And we're at this crucial point now where we have to decide, and that decision is going to be made quickly with the introduction of things like CRISPR. So the outcome of this project was a research project called Designing for the Sixth Extinction, very optimistic title, which was in the Science Gallery show, and it's now at ZKM in Karlsruhe, if you want to go and see it. And it's a huge photograph of a very perfect-looking forest. And as you start to look more closely, there are strange organisms lurking in the undergrowth. And this is one of the ones described in the gallery. Each is described in terms of a patent application. These are machines to save nature. And this is one of the questions that happened at this workshop, where people asked, could we use synthetic biology to infect nature to save it? And each was based on a real discussion happening in synthetic biology. So these are fictions, extrapolations into a future. But the idea that you could design kill switches so that biology would kill itself is something that we need to discuss. So for me, the ultimate problem that this question posed was this idea that if we industrialize biology and nature completely, will it still exist for us to save? So if we want to design a better future, I think we can use design to kind of open up this question about what that means and who's better is shaping our common future. And I think if we want to design living things, we have to ask, what kind of life do we want? So, we're all good. No time. Okay. Okay.
Time, so. In closing, you might have gathered from our conversations that design is chairs, but not only chairs and cars, and that science is not just abstruse papers and people working from an ivory tower. They're both about life, and together they can come to understand what a better future might be for all of us. And our intent tonight was to pollinate and to disseminate these ideas even more. So we thank you very much for listening, and we call back Steffi to introduce the next speaker. Thank you.